Hello, and welcome to today's joint webinar on implementing high-resolution, high-content image analysis in 3D spheroid models. I'm Judi Wardwell-Swanson from InSphero. I'm a senior application scientist, and I'm joined today by Gerd Heimlich from Yokogawa. So without any further ado, let us begin our webinar. So the objectives for today's webinar will include a brief review of how multicellular 3D models can help bridge the translational gap between preclinical and clinical drug development. We'll then discuss how technology platforms like high-content analysis can help researchers extract physiologically relevant data from 3D models. My co-presenter Gerd will discuss some of the special hardware and software considerations for 3D imaging and how Yokogawa is tackling 3D imaging challenges. We will then conclude the webinar with 2 case studies highlighting a few of the cell-level biological questions that can be addressed in multicellular spheroid models using high-resolution imaging and analysis approaches. So I suspect that a large number of you in the audience today have already implemented 3D models in your workflow, and some of you may already be reaping the rewards of that effort. Even so, it's not a bad idea to take a step back to ask ourselves 2 essential questions. What is the real motivation for implementing complex new processes in an existing workflow? And 2, what evidence do we have that will improve our drug discovery results? One answer to the first question is discussed on this slide, and it's related to the drug failure rates that are shown on the right-hand side of the screen. The bottom line is that drug approval rates are unacceptably low. And in spite of huge leaps in our understanding of disease mechanisms and a plethora of new drug modalities with superior specificity, the drug attrition rate has not improved appreciably over the past 20 years. Meantime, the costs associated with bringing a drug to market have nearly doubled over that same time frame. And according to the most recent Nature report, shown on the right, when they examined the failure rates for drugs entering the clinic between 2006 and 2015, 90% of drugs failed to reach the market due to either efficacy or safety issues. When a drug fails at this late stage, it's estimated to cost the pharma industry up to $2.6 billion. And it's important to remember that each one of these failed drugs underwent extensive preclinical testing before it reached the clinic, which suggests that there's a tremendous unmet need for improved translation. In other words, preclinical models that are better able to predict the human response. The image at the bottom of the screen shows our 2 most commonly used preclinical models, animal models and 2D cell culture models. And while each of these models provides important, indispensable information about compound activity and safety, each has an inherent flaw. Despite significant advances in humanizing mouse models, mice are still not humans. They don't develop the same diseases or respond to drugs in the same way as humans. And the most widely used 2D models are human, but they're not physiological, meaning they don't behave like integrated multicellular tissues, and in many cases they no longer resemble, at either the morphological, functional, or genomic level, the primary cell from which they were derived. In contrast, 3D human in vitro models exhibit a number of physiological features that can partially bridge these gaps. And on the next slide, I'll review a few of these features. There are many types of 3D models, including bioprinted, scaffold-assisted, organoids, and scaffold-free spheroids. For the purposes of today's talk, I'm going to focus on a very specific type of 3D in vitro model, multicellular 3D spheroids. 3D spheroids are very attractive models for drug discovery applications. Like 2D models, they are scalable, robust, and their uniform size and cellular composition leads to reproducible data. They're also amenable to a wide range of endpoints. However, this is where their similarity with 2D ends. For when you convert cells to 3D spheroid culture, they can acquire a number of more physiological features. First, they acquire a significantly longer functional lifespan in culture, sometimes up to a month or more without subculturing. Cells in 3D also take on a more native architecture. The cells inside a spheroid are in 360-degree contact with other cells. They adhere to each other through coherence that are expressed on their cell membranes. And this facilitates the formation of a very compact tissue, as shown in the immunofluorescent image of E-coherence staining in a mature liver spheroid on the right. Coherence also facilitates cell-to-cell communication, such that cells in 3D culture receive their cues from other cells rather than from the artificial surface. The result is that cells cultured as 3D spheroids resist dedifferentiation, and they retain their native morphology. As an example, primary hepatocytes that are cultured in 3D remain polarized, they form functional bile caniculi, and they stably express CYP450 enzymes for up to a month in culture. Compact spheroids also develop physiologically relevant molecular diffusion gradients for CO2, oxygen, nutrients, and waste products. These gradients in turn produce differential cell zones. Zonation has been heavily studied in tumor biology, where a proliferating, quiescent, and hypoxic zones have been described. These zones then correlate with areas of the tumor with differential sensitivity to drugs. And because of their tissue-like morphology, spheroids may also be useful to model drug diffusion and the efficiency of drug delivery vectors. The most physiologically relevant spheroid models, however, are comprised of all the cell types that form a particular tissue. And for the purposes of this talk, we will refer to them as multicellular spheroids. Multicellular spheroids can be constructed from the co-culture of different cell lines. They can be derived from human stem cells, or our personal favorite, they can be reconstructed from primary cells isolated from human donor organs. In addition to being multicellular, these spheroids can take on a remarkable resemblance to the original tissue. The coherence, which I mentioned earlier with regards to their role in cell adhesion, are also orchestrators of proper cell positioning within tissues. And the coherence present on isolated primary cells can enable proper positioning of cells in 3D models. This self-assembly or cell sorting process occurs during the spheroid formation and results in cell arrangements that are reminiscent of the native tissue. For example, as shown on the right, islet alpha and beta cells, labeled red and green respectively, form distinct clusters in the human islet spheroid. This organization is identical to the alpha and beta cell arrangement that occurs in native islets, and it facilitates tissue-appropriate paracrine signaling. Multicellular spheroids also more faithfully recapitulate native tissue at the transcriptome and proteome level. The heat map and PCA shown here is from a 2016 paper published by the Karolinska Institute. They compared the proteomes of native liver with 3D liver spheroids and 2D liver cultures. All were produced from the same donor material. The 7-day-old 3D liver spheroids from 3 independent donors cluster tightly together with the native liver samples, while the day 1 and day 7 2D cultures derived from the same donors form a very distant and distinct second cluster. The authors of the paper concluded that the 7-day-old 3D liver spheroids have nearly identical proteomes to native liver, whereas the 2D plated liver cultures have started to dedifferentiate in just 24 hours. But the ultimate test of how useful multicellular 3D models will be for drug development is the pharmacological validation. And one of the most comprehensive demonstrations of in vitro to clinical data translation that we are aware of is the 2017 Archives of Toxicology paper authored by Will Proctor from Genentech and Alison Foster from AstraZeneca. They examined 110 clinical compounds in human 3D liver microtissues and found that the liver model was twice as accurate at identifying drugs that cause drug-induced liver injury in the clinic than the 2D primary hepatocytes. So not only are multicellular 3D spheroids morphologically and functionally more similar to native tissue, but they can also respond to drugs in a more physiologically relevant fashion. Another feature that we like about multicellular steroid models is their versatility in terms of culture format. As shown here, the same 3D steroid model can be cultured in 96, 384, well, and even organ-on-a-chip formats. The latter format, organ-on-a-chip, enables an even higher level of complexity whereby researchers can study organ interactions as well as the effects of dynamic flow. Multicellular steroid models are also compatible with a large number of assay types, including many of the established clinical biomarker assays. And the opportunity to use the same biomarker endpoints for both the 3D in vitro model and in the clinic further increases the translational value of these models. Some of the assays that have already been implemented in 3D steroids are shown here. It's important to note that many of these assays were originally developed for 2D cell culture models or for large biopsy samples, and therefore may need to be modified or optimized for miniature 3D steroid tissues. The 3D compatible assays can actually be broken down into two general categories. So there's the global assays, those that measure responses at the spheroid level. These include secreted analytes as well as lytic endpoints. And then there's the high resolution assays, which measure tissue responses at the cell level. The latter are particularly well suited for multicellular tissues where the questions are commonly asked at the cell level. For example, in an IO application, scientists are frequently asked questions about the infiltration of specific immune cell types and subtypes of immune cells. In a type 1 diabetes, scientists are studying islet regeneration, are interested in evaluating beta cell specific proliferation in response to certain therapies. And there are also disease pathologies such as NASH, which involve the quantitation of subcellular and intercellular structures such as lipid droplets and collagen fibers. These questions require high resolution methods. Histology, single cell sequencing, flow cytometry, imaging mass spec, and confocal high content imaging are all good examples of high resolution assays. Of these, only high-content imaging also retains spatial information, in other words, where each cell was located within the tissue, and can be performed in a live format in order to collect temporal information. High-content imaging is also a relatively high-throughput platform, which it makes it quite compatible with scalable 3D models. High-content imaging would therefore seem like the perfect solution for physiological data, for extracting physiological data from multicellular spheroids. But anyone who's tried to image and analyze spheroids will tell you it's not quite as easy as it sounds. There are a number of things to consider before deploying a 3D imaging workflow in your organization. The first is the 3D model, which for lead discovery applications needs to be robust and scalable. And the second is the 3D culture plate, because the type of plate can make a significant difference in the success or failure of your 3D imaging assay. Ideally, you'd like a plate that is ANSI SLAS compatible and is therefore compatible with automation and high content imaging instrumentation. It should have a well geometry that minimizes unintended aspiration of the spheroid during the processing steps. It should have black walls and a thin optical bottom for enhanced imaging quality and speed. And last but not least, a small observation window will facilitate accurate spheroid identification. The Acra 384 well plate, part of InSphero's 3D microtissue platform, is a good example. So to summarize the first part of the presentation, a 3D platform starts with a physiologically relevant but scalable 3D model. It includes a plate technology that's compatible with downstream applications, in this case high-resolution imaging. And finally, a 3D platform often requires optimization of assay protocols for 3D models. And when pairing a 3D model with high-content screening endpoints, an image and image analysis platform that's fully optimized for 3D models is also highly desirable. A confocal instrument capable of high-resolution 3D imaging can be a significant investment for any organization, and therefore the imaging system selection should not be taken lightly. Now I'm going to turn the presentation over to Gerd from Yokogawa, who will discuss some of the features of their confocal imaging system, which makes them compatible with high-resolution imaging of multicellular 3D models. Gerd?
Hello everybody, thank you for joining today's webinar. My name is Gerd Heimlich and I'm the Sales and Marketing Manager for the Yokogawa Live Innovation business in Europe. Today I will present how our high-content imaging products can benefit your research in regard to three-dimensional live cell imaging in drug discovery applications. In the recent years, more and more researchers are moving from the classical two-dimensional cell models that have been used for decades to three-dimensional cell models in drug discovery. Three-dimensional cell models are also referred to as microtissues, spheroids, or organoids, and they have improved in morphological characteristics because they are mimicking the real in vivo tissue environment in a much better way. With that, three-dimensional models are of higher physiological relevance. As my collaborator Judi from InSphero just explained, three-dimensional microtissues can be easily handled with the proper equipment, displaying a long lifespan in culture. Most important, three-dimensional models can be adapted to validated cellular endpoints in drug discovery. So in summary, researchers are equipped with a highly relevant cell model that should better predict the potential effects of drug candidates onto a given cellular environment. Nevertheless, where there is light, there can be shadow, and there are certain technical challenges with three-dimensional cell imaging. To obtain volumetric information, multiple images along the sample z-axis have to be taken, exposing the cells to increased laser energy, which might cause phototoxic effects on the sample. Therefore, it is beneficial to use an imaging instrument allowing short exposure times. Additionally, light penetration and distribution in three-dimensional cell structures can be unfavorable or uneven, resulting in suboptimal signal-to-noise ratios. For that reason, the imaging instrument should be equipped with a sensitive camera with a wide field of view. Because of the increased complexity of the cellular architecture in the three-dimensional versus the classical two-dimensional model, more image planes have to be acquired. Instruments with improved focus routines help the researchers to obtain an optimal sample view in a shorter time. Usually, there is only one spheroid per well. If the imaging would be done at high resolution for the whole well, the acquisition time would be extremely long, resulting in many empty or blank images. Most effectively is the object of interest identification at a low resolution first, followed by the final image acquisition at higher resolution. Last but not least, once all images are collected, the researchers need a powerful analysis software to convert the Z-stack image information into meaningful volumetric data. In either two-dimensional or three-dimensional high-content imaging, we look at a very general workflow. The image plane with the cells of interest is acquired, and from this image plane, the analysis software identifies countable objects of interest. Here, in more and more applications, artificial intelligence supports the users. Those objects are counted and graphically displayed to address the researcher's question. The object of interest can simply be fluorescence intensity, but also count of objects like nuclei or mitochondria, or the whole volume of a cell colony. Yokogawa offers two high-content analysis platforms, a high-throughput device with multiple cameras and a kinetic module, which we call the Cell Voyager 8000, as well as a benchtop instrument, which is called the CQ1. The CQ1 instrument was actually used in our collaboration with InSphero, and I will talk about its specifications in just a few minutes. We complete our high-content analysis portfolio with an analysis software suite, which we call CellPathfinder, and a data management infrastructure, which is beneficial in high-content analysis applications, but it's not in scope of today's talk. In our Yokogawa high-content imaging instruments, we are using proprietary technology that is based on dual spinning disks. In most classical confocal microscope, a single high-power laser beam scans the whole imaging plane, exposing the cells to increased energy levels, potentially causing photobleaching or phototoxic effects. In contrast, in the Yokogawa dual spinning disk instrument, The laser is split by the Nipkow pinhole disk into about 1,000 moderate energy laser beams, and those scan the whole sample in much faster, avoiding any significant phototoxic impact. Additionally, a second microlens disk maximizes the laser energy in each pinhole, allowing maximal excitation even in deep tissue layers. In our Yokogawa high-content analysis system, we combine dual spinning disk technology with stage incubators, generating an imaging instrument that allows high throughput acquisition of high-quality confocal images of living cells. The light distribution and penetration is effective even in thick three-dimensional microtissues, and multiple Z-stacks can be acquired without phototoxic harm to the sample. The implemented stage incubator tightly controls, respectively regulates temperature, CO2, and humidity levels. With that, the three-dimensional cell structures can be kept alive for hours up to days in the incubator chamber. This allows what we call four-dimensional imaging, meaning the analysis of a three-dimensional microtissue during a long-term time-lapse experiment. Based on the unique technical specifications, the Yokogawa CQ1 benchtop high-content imaging instrument allows scanning of a 96 well plate in about 2.5 minutes. Besides the dual spinning disk technology I introduced already, the CQ1 is equipped with a sensitive S-CMOS camera with a wide field of view. That even allows acquisition of a 250 micrometer spheroid in a single field of view using a 20x objective. Up to 6 objectives can be exchangeably, and 4 lasers of the indicated wavelengths can be integrated. The CQ1 offers a hardware laser-based autofocus, which is of importance for the acquisition of three-dimensional microtissues, as mentioned at the beginning. The high accuracy and high repeatability of positioning with the XY stage in the CQ1 instrument makes it possible to perform map images and multi-point acquisitions. On top of the actual acquisition platform, Yokogawa offers an analysis software suite which we call CellPathfinder. This software offers a basic and advanced mode to be tailored to the varying knowledge levels of potential users. CellPathfinder contains a variety of predefined protocols for typical questions in the context of high-content imaging, respectively cellular analysis. In regard to our topic of the day, CellPathfinder contains a three-dimensional analysis menu option, which makes the image data analysis of spheroids very user-friendly. With the following examples, I would like to introduce you to some special features of CellPathfinder. Starting from the sample image, our analysis software detects the analysis of interests according to the set specifications by the user. The actual object counts can be displayed in various graphic formats like heat maps, scattergrams, or histogram plots. One of the unique features of CellPathfinder is the direct link of a particular dot in the graph, for example an outlier, to its corresponding cell image by simply clicking on the dot. Also, for a time course experiment, it is possible to gate defined populations and observe the change of these populations in this corresponding pie chart diagram over time. I would like to end my part of the webinar with a very short example of a three-dimensional microtissue time course experiment. As you can see in the displayed graph, the growth of an embryonic stem cell colony, as well as the temporal changes of the expression of a marker for undifferentiated cells, were observed over a time course of 48 hours. All nuclei are displayed in red, while the undifferentiated cells are displayed in green. The expression level ratio, respectively expression amount, for nano EGFP were analyzed for the customized outer and inner region of the stem cell colony. The growth of the colony and differentiation status can nicely be displayed over the time course of the 2 days. With that, I'm at the end of my part of the webinar. I thank you for your participation and attention and would like to hand it over back to Judi. Thank you.
Thank you, Gerd. And for the final part of today's presentation, I would like to share 2 real-world examples where high-resolution, high-content imaging was instrumental in extracting physiologically relevant data from complex multicellular 3D models. The 2 case studies I'll share today involve the characterization of steatosis in a 3D liver spheroid model and the characterization of immune cell infiltration in a 3D tumor spheroid model. The first example involves the quantification of lipid droplets for steatosis in a NASH liver disease model. Steatosis is an important hallmark and an initiating event in the development of NASH. For this study, we used InSphero's 3D Insight liver model, a multicellular model comprised of human donor-derived hepatocytes, Kupffer cells, liver endothelial cells, and human stellate cells. To simulate healthy and steatotic liver, the 3D liver model was exposed to two media conditions, either lean or NASH. Under the lean condition, any intracellular lipid accumulation is the result of de novo lipogenesis. The NASH media, however, contains a mixture of free fatty acid in addition to higher levels of glucose and fructose. In this case, the lipid accumulation is primarily a result of free fatty acid uptake. There were three biological questions we wished to address with this study. The first was, what is the level of lipid accumulation under lean and NASH conditions, and how does it compare to the clinical manifestation of the early stages of NASH? And the second question was how the clinical compound forsocostat, an inhibitor of ACC, which is the rate-limiting enzyme in fatty acid synthesis, how does it affect lipid accumulation under these two conditions? And then we further wanted to discriminate and classify lipid accumulation as either microvesicular or macrovesicular steatosis. And we were interested in this last point as the presence of microvesicular steatosis can be used in the scoring and clinical assessment of NASH severity, and it's also thought to be a prognostic indicator. So following a 10-day treatment in either the lean or NASH conditions, the liver steroids were processed for high-content imaging. The lipid droplets were visualized with Nile red and the nuclei with DAPI. A 40x high NA air objective was used to acquire Z-stacks for each treatment group. And the Z-sections were acquired at 4 micron steps to a depth which was roughly equivalent to the center of the steroid. The images in the top row are maximum intensity projections showing the lipid droplets in liver steroids cultured under either the lean or the NASH conditions. And the lean is shown on the top left and the NASH conditions on the top right. The corresponding images in the bottom row illustrate the image segmentation or the identification of the individual lipid droplets. Of the features that were extracted during the image analysis, lipid droplet count was calculated for the entire acquired volume, and then it was compared across the 4 treatment groups, the lean condition, the NASH, and the NASH condition, either with or without the ACC inhibitor. Lipid droplets are present in the liver microtissues under both lean and NASH conditions. However, a significant increase in lipid droplet number is observed in the NASH condition, and that's shown in the first and third bars in the bar graph on the right. The ACC inhibitor forsocostat reduces the number of lipid droplets under both conditions, and that's shown in the second and fourth bars. Not surprisingly, forsocostat has a more robust effect on lipid droplets that are generated via de novo lipogenesis than those that are generated from the uptake of exogenous free fatty acid. Further, we were able to classify the lipid droplets into 2 size groups, those that were greater than 30 cubic microns and those that were less than 30 cubic microns. In the lean condition, forsocostat appears to favor the reduction of lipid droplets that are larger than 30 cubic microns, or the macro droplets. And that's shown by the decreased size of the orange wedge in the right-hand pie chart. The results of the study suggest that we can effectively quantify subcellular lipid droplets in a 3D steroid model, and that we can potentially further characterize the NASH phenotype by the extent of both microvesicular and macrovesicular steatosis. Ultimately, this type of information could then be compared directly to clinical histopathology data. The second example is an immuno-oncology case study and involves the evaluation of immune cell infiltration and subsequent tumor killing. In this study, both a spatial and temporal analysis of individual cell populations was examined in order to quantify both the kinetics and depth of the immune cell infiltration, in addition to quantifying the kinetics and efficiency of the tumor spheroid destruction. We used a tumor co-culture model that was comprised of both epithelial and stromal cells, and we evaluated 3 conditions: tumor spheroids without immune cells, with native immune cells, with naive immune cells, or with 3D CD3, CD28 activated immune cells. As the illustration on the top shows, the immune cells are added to fully formed tumor spheroid co-cultures. And following the addition of immune cells, the infiltration and tumor cell killing was monitored for 2 days in a live imaging mode. The biological questions we were interested in addressing with this study were: how do the naive and activated immune cells affect the tumor spheroid volume? And can we quantify the kinetics and depth of the immune cell infiltration and learn something about the fate of the infiltrated immune cells? For this study, the tumor cells were labeled with a green probe and the immune cells were labeled with a red probe. We acquired confocal Z sections in both channels and at regular time intervals for a total of 39 hours. By analyzing the total tumor cell volume and the spheroid-associated immune cell populations independently, we obtained some very interesting insights. As expected, untreated tumor spheroids show a slow, steady increase in total tumor volume over time, and that's shown by the blue trace in the top graph. In tumor spheroids that are treated with naive PBMCs, shown by the green trace, we observe a rapid but minor decrease in tumor volume, which lasts for approximately 4 to 5 hours, and it suggests a certain level of innate immune response by non-HLA matched PBMCs. In contrast, CD3, CD28 activated PBMCs, the pink trace, we observe a prolonged and highly efficacious tumor killing profile that's consistent with T cell activation and which results in complete tissue destruction by 20 hours. Interestingly, when you look at the PBMC population by itself, as shown in the middle graph, we see a very sharp increase in the total number of activated PBMCs at the spheroid site during the first few time points, which peaks around 4 hours, and that's shown by the pink trace. And then it slowly drops off, while naive PBMCs show a more gradual accumulation at the spheroid site, which plateaus by approximately 9 hours and remains constant for the duration of the study, and that's shown in green. The sharp peak in the activated condition suggests that spheroid-associated PBMCs are either increased in their migration into the spheroid or that a rapid expansion of activated immune cells is occurring within the tumor spheroid. The subsequent slow decrease in spheroid-associated PBMC number corresponds then to the destruction of the spheroid. And lastly, each immune cell was annotated with X, Y, and Z coordinates, which allows us to determine where each immune cell is within the spheroid at any given time. The X and Y axes in the centroid maps on the bottom of the slide correspond to the X and Y position of each immune cell within the spheroid volume. The color coding, however, corresponds to their relative Z position within the spheroid. So blue-green coloration corresponds to sections of the spheroid that are closest to the bottom of the plate, yellow-orange corresponds to the central sections, and red corresponds to sections at the top of the spheroid. At the 4-hour time point, we observe that most of the naive PBMCs are at or near the bottom of the well by the predominantly blue-green coloration, and that they are more concentrated near the periphery of the spheroid. This pattern is consistent with cells surrounding the spheroid rather than infiltrating. Whereas the activated PBMCs are distributed evenly throughout the spheroid in all 3 dimensions, and that's shown by their distribution in the XY dimensions, but also their blue, green, yellow, orange, and red coloration. This pattern is consistent with full immune cell infiltration by 4 hours. Now that we've demonstrated the feasibility of conducting 3D in vitro IO studies in real time, our next steps are to examine the infiltration of specific immune cell types that either mediate or suppress tumor cell killing. Ideally, this would include modeling of antigen-specific immune response or adaptive responses, and ultimately we plan to use these 3D tumor models, as well as 3D tumor models derived from patient tumors, to evaluate IO therapeutics and combination therapies in 3D in vitro studies and in real time. So to summarize today's webinar, multicellular steroids represent a significant step in the direction of more physiologically relevant models for use in drug discovery applications, and they fulfill this role without departing from the requirement of scalable, robust, and reproducible in vitro models. Looking towards the future, we foresee opportunities to further increase the physiological relevance of these models by combining several different tissues on our AcroFlow organ-on-a-chip platform. For example, we're currently working on a more systemic model of metabolic disease by combining liver and islets. In our experience, multicellular 3D steroid approach works well for a large number of tissue types. A few examples of the functional multicellular steroid models we produce at InSphero are shown on the right. However, at the same time, we recognize that more advanced tissue engineering approaches may be required for complex organs, such as kidney. We also believe that high-resolution technology platforms, such as high-content imaging, can help researchers exploit the full potential of multicellular 3D models by enabling evaluation of events not only at the spheroid level, but also at the cell and subcellular levels. So if you enjoyed today's presentation, I hope you will take a look at InSphero and Yokogawa's joint publication that's in the current issue of SLAS Discovery, where you can find a more in-depth discussion on implementing a 3D workflow. I'd like to acknowledge the folks at InSphero that generated the beautiful data that I showed today, as well as our collaborator, Mahomi Suzuki at Yokogawa, who provided us with exceptional training on the Yokogawa QC1 and also performed a significant piece of the image analysis. So this concludes today's webinar on high-resolution, high-content imaging of 3D steroid models. Gerd and I would like to thank you for your attention today. You can find both of our emails at the bottom of the slide, and we welcome you to reach out to us if you're interested in learning more about how our respective companies can help you get started with either 3D models or high-resolution 3D imaging. Thank you very much.